Good day, this is Professor Will, CBMD PhD. Hey, Sunday, March 24, 2019 is 11:30:42 Eastern Standard Time. And this is a brief description on Aspergillus. So let's get started here. Aspergillus is a genus of fungi widespread in nature that includes almost 200 species, several of which cause disease in humans and other animals. Species of Aspergillus also play important roles in industrial microbiology and food production. Despite the value of Aspergillus in industry, this microorganism can also cause trouble in food and other product manufacturing when it contaminates formulations. Aspergillus is a ubiquitous microorganism found in natural non-extreme environments. The fungus exists in any place supplied with organic matter. Some moisture and moderate temperature range of 50 to 120 degrees Fahrenheit, 10 to 50 degrees Celsius. The outdoors and indoors both provide favorable conditions for Aspergillus, although it lives as a normal inhabitant of soil. It also readily enters the indoors when breezes blow fungal spores inside. Aspergillus grows in cool, moist places inside buildings and can affect human health at high concentrations by causing allergies or other more serious ailments. In the controlled conditions of laboratories, Aspergillus has become a common focus of study. Characteristics of Aspergillus. Aspergillus is one of several fungi more commonly known as molds. Molds are multicellular fungi that grow in a visible, fuzzy or fluffy colony. This fuzziness results from the growth of mycelia, which are bundles of long branching filaments that intertwine as they grow. Aspergillus also produces spores, which act as the means for dispersal and reproduction. Spores travel through the air. This mode of transmission is called airborne transmission. Eventually the spores settle out of the air onto a surface where, if organic matter, nutrients, and moisture are present, they germinate within 2 to 3 days. Spore germination entails a process of growth from the spore form of a microorganism. Fungi such as Aspergillus germinate from fungal spores. Endospore-forming bacteria also germinate. Also, the term spore has been used for both fungal structures and bacterial endospores, such as those of Bacillus and Clostridium. These two different types of spores are unlike. Upon germination, Aspergillus spores multiply and form colonies that appear fuzzy and black or greenish. The mold that grows on spoiled fruit in the refrigerator provides a familiar example of Aspergillus growth. Aspergillus also grows on non-food items such as leather, clothing, bathroom tiles, and carpets. The mold's favorite conditions are cool, dark places with high humidity and little airflow. Closets, cellars, and attics serve as common indoor habitats for Aspergillus. Fully germinated Aspergillus contains structures typical of molds, mycelia, and hypha. Mycelia are long, stringy filaments that grow into the familiar tangles or fuzz that people associate with most molds. Each mycelium is composed of bundles of individual hypha. A single hypha is string-like growth of about 2.5 to 8.0 micrometers in diameter. Aspergillus hypha contain characteristics that are not found in all molds. For example, their hypha are called septate, meaning structures that contain distinct sections separated from one another by cell walls. Aspergillus hypha also tend to grow in an upward direction, which gives colonies their fluffy appearance. Hypha may also grow an appendage called conidiospore, which extends outward or upward from the main hypha structure and supports a round end or head. This most distal part of the conidiophore holds hundreds of small 2-5 micrometers in diameter. Conidia, an asexual reproductive component. The conidia are usually recognized and referred to as mold spores. Aspergillus is a pathogen. People and animals inhale airborne Aspergillus spores into their respiratory passages. This route often begins an infection or an allergic reaction in the respiratory tract. In rarer cases, inhaled spores lead to infections of organs in addition to the lungs or other parts of the respiratory tract. Members of the Aspergillus genus cause a group of diseases collectively known as aspergillosis. Aspergillus mold often acts as an opportunistic pathogen, which is any microorganism not normally dangerous to the body but able to cause infection under favorable conditions. For example, people who have weakened immunity are more vulnerable to opportunistic infections than healthy persons with strong immune systems. Certain members of the population compose a subpopulation that is at higher than normal risk of contracting opportunistic infections, such as the following groups: the elderly, young children and infants, pregnant women, people with long-term chronic existing disease, people with acquired immunodeficiency syndrome, AIDS, chemotherapy patients, or organ transplant recipients. Aspergillosis has been further differentiated into the 4 following varieties: non-invasive, bronchopulmonary aspergillosis ABPA, predominantly an allergic reaction within the bronchia of the lungs, especially in the asthma sufferers. Invasive pulmonary aspergillosis, IPA, attacks one or more of the body's internal organs after spreading from the lungs. Chronic pulmonary aspergillosis masses of mold cells grow and cover much of the inner surface of the bronchia. Skin aspergillosis, infections of the skin. Opportunistic Aspergillus infections most commonly cause irritation to the sinuses and upper respiratory tract. Deeper penetration of the respiratory tract results in ABPA, in which both asthma and non-asthma sufferers develop allergy to the mold spores. Invasive infections of IPA also start in the lungs. When either the fumigatus or A. flavus infects the lungs, the mold typically does not invade the bloodstream, but instead grows directly on the lung tissue. A massive mold growth called an aspergilloma may cover the bronchial sacs and tubes of the lungs. This condition makes breathing very difficult by reducing the infected person's lung capacity. The volume of air a person can inhale with a single deep breath. If Aspergillus escapes the lungs and invades the bloodstream to cause IPA, the microorganism infects other tissue or organs and can lead to death. A. fumigatus and A. flavus have also been implicated in an ailment called allergic aspergillosis, which develops within 1 to a few minutes after a person inhales spores. In these cases, a person may experience difficult breathing or more serious reactions such as asthma. Allergic aspergillosis has been linked to sick building syndrome, SBS, which is a situation in which building occupants develop health problems related to indoor air of poor quality. This circumstance is also called building-related illness. Chemicals in addition to microorganisms have been blamed for SBA, but finding the exact cause presents difficulties. Hundreds of chemicals, particles, pollen, molds, and other substances in the air may contribute to SBS. For this reason, experts on indoor air quality, IAQ, have not reached agreement on the extent to which Aspergillus or other molds contribute to SBS. A 2008 update on SBS in Business Week illustrated the reason why this illness has challenged physicians and microbiologists. There are a number of indoor air pollutants that contribute to poor IAQ and the spread of airborne disease. These include biological contaminants such as molds and bacteria, and combustion pollutants like carbon monoxide and toxic particles. Even the building itself is a factor, since toxic substances emitted from building materials and furnishings degrade IAQ. Aspergillus normally causes no harm on the skin and only poses a threat of infection if the skin receives cuts, burns, or similar wounds. Aspergillus species, especially A. fumigatus, can be common in decaying vegetation, so people who tend compost piles, gardeners, and farmers may have a higher risk of exposure to the spores. Hi, this is Professor Will, CBMD, PhD. Today is Sunday, March 24, 2019, and this is the end of my brief description on Aspergillus. Hope you enjoyed listening. Have a great day.